The Zoom is working, so some people are zooming in. And today we have a presentation by Matt Engel. We are very fortunate again to have a presentation by a company, which is a good sign that this field is becoming more mature and real technologies find real applications. So Matt is a scientist and entrepreneur. He did his bachelor degree in Carnegie Mellon and then PhD in University of Heidelberg, actually Max Planck Institute next to University of Heidelberg, and then postdoc in Nick Mellish's lab here at Stanford. And right after postdoc, and that's a Stanford tradition, people start companies, so Matt started Paradromics. And I thought that the first application was to read thoughts and restore speech after stroke. But here's maybe the...
对，因为我要听一下这个报告。
Wow, question started.
哦，你感兴趣吗？
Okay, Karina. No, I'll ask her to turn them off. Okay. Anyway, so
其实在楼上，他正好有Zoom，我就懒得上去。
We have competition.
你要听，我可以把链接发给你。
So yeah, so the Matt will tell us today about the technology that Paradromics is developing. Can you mute them off? Some people on Zoom speaking. And yeah, so the podium is yours.
就很大的标题，对不对？对。一般这种公司人讲东西就很火。
My hands are a little bit tied given my position, so I won't be talking about everything that Paradromics is doing, but I will be talking about the things that we either have made public already or the things that will become public in the next 2 or 3 months. So it's a little bit of a preview, but there will still be certain things that we're doing that I leave out of the presentation. I'm sure you understand. We got started in Nick Mellish's lab looking at how to scale up in vivo recording, specifically in vivo electrical recording, taking large arrays of microwires and bonding them onto CMOS ASICs for high data throughput BCI. And when we left, at the time there really there was no hardware to pull down thousands and thousands of channels simultaneously from a large format microwire array. And so that was the first thing we built at Paradromics. And so the first thing that I'm going to tell you about is what we call the Argo system. It is essentially a high-speed camera that can go up to 39,000 frames per second. It has 256 by 256, 65,000 pixels, and each pixel is a voltage amplifier. So you can bond arrays of microwires on there, and they can be very large format. This gives you a little sample of the specifications, looking less than 10 microvolts peak-to-peak RMS noise in the band that you would want for LFP and spiking, arrays practically going up into the tens of thousands of channels. So first, to give you a little bit of an idea of what the system looks like, we have on one hand the array component, these bundles of microwires. The bundles of microwires are press bonded onto a CMOS ASIC, and then the CMOS ASIC is read out by electronics that effectively mirror a high-speed camera. We think that microwires is a really good starting point for neural recording because it's the kind of thing that can translate really well into a human BCI. Microwires have been around for decades. They're known to last a very long time in vivo. When you make them small enough, they're minimally invasive. We have a lot of existence proofs for microwire arrays being used in humans and primates on the period of years. They're made of materials that last a very long time, and when they do fail, we know the material and biotic causes of failure. So it's a place where we can do engineering. One of the downsides of microelectrodes, as they're currently used, is that they're not very scalable. Typical sort of lab scenario is that you're hand sharpening tungsten microwires, you're assembling them in an arts and crafts project into an array of maximum 100, and then you're hand soldering each of those wires onto a discrete lead to connect to your readout electronics. And, but this is exactly the kind of thing that you can engineer around, and I'm sure you can appreciate that. More fundamentally, small microwires can insert into tissue and do minimal damage. And this is just an example of a paper, Thelen et al. From 2011 that looked at microwires that were floating in the brain, i.e. not connected to the skull and able to move with the brain. And it asked the question, does the size have a strong impact on the tissue response? Answer, yes. 200 micron diameter wires cause quite a bit of glial response and astrocyte response, and you lose quite a number of neurons in the surrounding area. When you drop that diameter down to 50 microns, the result is that you lose fewer neurons and you have less immune response in that area. And when you drop the electrode size less than 50 microns, we have found that the benefit continues. And I just have an example of a video here demonstrating one of the principal reasons why having a smaller wire is better. This is an example of a fluorescently labeled 20 micron microwire being inserted through a mouse brain, and the vasculature is dyed red. And you can see that there's a kind of characteristic mesh size of the vascular network around 50 microns. When things are smaller than that, they tend to insert without doing a lot of damage to the blood vessels, which means one, you have better oxygen supply in that area, and two, you have fewer inflammatory cytokines pouring into that area, which are predictive of bad long-term outcomes. We're specifically using sharpened probes. I think that's obvious to most people. Sharper things go in more easily. Sharpening has traditionally been something that's hard to scale, but I'll show you a video of a kind of production of a lot of microwires at scale in the next couple of videos. So here we have some 20 micron wire. You can order it commercially. We're passing it through an annealing setup to make it straight. It's important when you're inserting things in the brain that the wires be very, very straight, otherwise they buckle when you try to insert them. Here we're respooling the wire onto a template for coding. Laser cutting them. So now we have an array of 65 wires, and we're sharpening them in parallel. These are platinum iridium microwires, 20 microns. We have a pipeline now. I think that we're producing something like 20 or 30,000 wires per week when we're up and running, which is enough to make some pretty large arrays for testing. We have the process quite reproducible now. We can make tips of a reproducible diameter. We can control the taper reproducibly. These are probably boring things for Stanford, but if you want to make tens of thousands of these and have them insert correctly, you really have to have a reproducible control over every step in the process. The next step, once you have these arrays of sharpened wires, is to do a chemical vapor deposition to protect the wires. Then we assemble them all into a single block. These assembled bundles, we can now make up to arrays of 50,000 wires. And a lot of this processing is similar to the work that Nick is doing here at Stanford. Polish the bundle flat so that it can be bonded to the CMOS. And then we're etching back a lot of sacrificial material to reveal the sharpened wires. And that's a plasma etch. Here we can see the bundle pressing down onto the CMOS chip. So this particular bond is just a pressure bond. For implantable electronics, it's useful to use something like solder bump bonding, a more permanent solution. But this allows us to rapidly iterate on the front end while we test different designs. The individual sensors are effectively voltage amplifiers. We work together with a Belgian CMOS company, Cailesti, that manufactures sophisticated and state-of-the-art image sensors. And so we work with them to build this voltage sensor array with 65,000 channels. It's read out at full frame. I think that's one thing that differs significantly from a lot of devices you might have seen in the CMOS MEA literature. It's common for people to say, oh, I have 100 billion channels, but the readout electronics allow you to select a subset of 5 channels at a time. But this system reads out full frame at 12 bits, 39,000 samples per second, 65,000 channels. So you can get, indeed, a lot of data. I think the system clocks out at between 24 and 30 gigabits per second. To read out all of that data, we have some specialized electronics. We have ADCs, high-power FPGA that then sends the data over a 40 gigabit per second optical interface to a computer that can record all of the data to disk. So let me show you a little bit of some of the data that we've collected with the system. This is an example of 1,200 inserted electrodes into a rat brain. Here's the insertion. I normally don't get to show these kinds of videos in my talks to venture capitalists, so I'm happy to show you all the surgeries. This is a video that I think will play now of 1,200 wires, I think something like 800 spiking neurons. So every point that you see here is a spike rate. The spiking is pretty sparse, so most of the time it's just one spike per bin. I think the bins are 20 milliseconds or something like that. So this is some of the data that we pulled off of the system. A little bit of a summary of what this looked like across the active channel map. You'll see that the spike yield was relatively low. When we're doing acute head-fixed recordings, it's really hard to do high channel count acute head-fixed recordings with an array like this. Some of you are familiar with the Utah array, know that the yield when you insert the Utah array initially is almost always 0. You cause a lot of inflammation when you first insert things. Takashi Kozai at the University of Pittsburgh has looked pretty specifically at some of the responses, looking at calcium imaging before and after, and sort of for a period of hours following insertion. And so it's pretty common in this kind of format not to see high spiking yields right off the bat. And that's also consistent with some of the work that Nick's lab has been doing over the last months that show that when they do these chronic microarray bundle insertions, the initial yield is low, but then it recovers after a period of weeks. So despite the system is very interesting, but some of the acute results that come back actually aren't that interesting in my view. This is an example of, again, a raster plot of the video that you just showed over a period of a few hours, I think, or actually maybe minutes. Rats and mice are not a very good translational system for developing human-facing BCI. We do all of our work in sheep. We're actually working on a piece that'll come out, I think, in the next month or two, specifically about sheep as a translational model. It has a lot of advantages over non-human primates in terms of the iteration time. It's not feasible to do many non-human primate experiments in the scope of a few weeks. But when you're doing this kind of device design, you need high iteration. At the same time, a lot of the system level considerations for designing a BCI for humans, they don't translate well between mice and humans. The anatomy is very different, the thickness of the pia, the surgical implantation method. So I think in general for the whole field, it's important for people to be cognizant of the models that they're working in. I think a lot of work goes into rodents for obvious reasons because you can prove out concepts quickly there, it's cheaper. But in terms of human-facing BCI, there are often translational issues. And similarly, a lot of the most exciting work, see Paul and Krishna in the audience, a lot of the most exciting work in BCI happens in non-human primates. But from a translational perspective, from developing human-facing BCI, that's often not a good place to iterate. Just pause to make that point here. So we've been doing a lot of work in sheep for the reason that the acute insertions don't often look very good, and our system is by nature a head-fixed anesthetized recording system. We've been collecting a lot of data more recently using micro-ECOG arrays that we can also bond onto our Argo system. So this is an example of a 30,000 channel micro-ECOG array with 60 micron pitch that we use to record responses in the auditory cortex of a sheep. Here's just a coarse summary of some of the data that we've collected, looking at kind of baseline power, power evoked by speech data sets, and then post-euthanol, which essentially means the animal is not alive. Does baseline control to look at the power. We've done things like looking at peristimulus time histogram, looking at the response across channels to pink noise stimuli, looking and seeing that most of the channels are noise responsive on a pixel by pixel basis, constructing tuning curves. This is kind of basic sensory physiology. We've also looked across the entire bundle. Here, the color coding, each color represents a frequency, with red being lower frequency, blue being higher frequency. The intensity of that frequency is the statistical significance of the response to that frequency. And so what we've created here is essentially a tonotopic map in the sheep based on the micro-ECOG data. It roughly mirrors what we would expect to find based on other higher animals. There's not a lot of physiology that's been done in sheep, so many of the results that we get in the lab are the first results in that model system, so we often have to validate that. I'll show you an example of, this is, I believe, 24,000 micro-ECOG electrodes collected on the Argo system. Each dot represents an amplitude. It's filtered in the gamma range, and this is the sheep's response to human speech data set. So these recordings that you're looking at now are, to my knowledge, the largest in vivo electrical recordings ever made. It's quite fascinating to me. And here's another way of viewing that data that includes the full band activity in the white wireframe, and then the gamma band activity is colored and superimposed over top of that. So one of the things that we've been working on a lot is looking at different ways to visualize these data sets, since sometimes when you're collecting data at very large scale, even just getting a sense of what you have in an instant can be more challenging than smaller sized data sets. One of the things that we were very interested when we collected these recordings was knowing whether we were oversampling collecting ECOG data at 60 micron pitch. Some people, including myself, thought that was redundant. I thought there wouldn't be much additional information there. We used a technique called shared variance component analysis, which is a little bit similar to PCA, but you can think of it as kind of a cross-validated PCA to determine whether and it essentially asks the question, what is the dimensionality of the data, and is that dimensionality reliable or spurious? And so we were using this dimensionality as calculated by SVCA, and we're asking the question whether the predicted underlying dimensionality of the system scales as a factor of the number of channels that we include in the analysis. When we include a subset of channels, we see that the dimensions that we recover are lower, and as far as we extended the analysis out, as many channels as we had, we saw that the predicted dimensionality was higher. So on first blush, it looks like there is meaningful data contained in the larger data sets, but this is very much kind of a zeroth order analysis. And then each of these colors represents a number of different stimuli, going from sort of baseline as the ongoing spontaneous activity, and then PT 1 through 5 represent data sets with pure tone stimuli ranging from 5 tones to about 50 discrete tones. And then the pink line is the human speech data set. So we do see a trend. We see a trend that the dimensionality that we recover from this does reflect the complexity of the underlying stimulus that we fed into the system, which is consistent with what we would expect. I have to admit, though, this isn't my strength, and this is really kind of a first blush analysis to make sure that we're getting meaningful data from such large channel count. The other thing that I'll talk about a little bit, and I think we'll be putting a paper out about in the next couple of months, will be the compressive sensing that we're doing. When we talk about the Argo system that collected the data that I just showed is about yay big, that's obviously not suitable technology for a brain implant. A brain implant, we want something in the form factor of, say, the Utah array, a few millimeters by a few millimeters. All of the discrete components and all of the electronics in our system can be scaled down into a single ASIC. The technology for implanting active ASICs in that sort of form factor exists in the form of co-fired sort of metal ceramic composites. You can build hermetic boxes to fit everything. But one of the things that doesn't scale down is power dissipation. Our current system dissipates about 20 watts of power. And if you shrink that down into a small form factor implant, you cook the brain. And the biggest sort of power challenge associated with trying to do high bandwidth in a small volume is that you're either in a situation where you're digitizing in situ, in which case you're fighting an industry trend that you're not likely to improve as a neuroscientist, which is the power per conversion step in analog to digital converters. I know that Boris has been tracking that for, he's been tracking that for probably 15 years or something like that. And it's not going to get better by anyone likely in our company. And so if you want to try to push more data through the pipe, the answer is not to try to develop better ADCs, in 6 months, nor is the answer to try to transmit the raw analog signals, because in order to have a good signal to noise, you'd have to have a very high amplitude analog signal, and the signal will scale, the energy required to do that transmission will scale quadratically with the amplitude of the signal. So again, you'd be fighting a bad scaling rule. And so the result is that you can't either digitize everything in situ or transmit out the analog values unprocessed. But what you can do is you can change the paradigm a little bit. Instead of doing all of your processing in the digital domain, which is where compression would normally occur, you can do your compression in the analog domain. And so I'll show you just a brief schematic that demonstrates what we're doing. Essentially, it comes from the realization that calculating principal component scores from a set of eigenvectors is essentially the same as convolving your input signal with those eigenvectors, with those bases. And the convolution operation can be easily implemented in the analog domain by a complex linear filter. And so we've baked into each pixel of our system an analog filter whose frequency response is the eigenvector, is the principal component that we've calculated offline. And so we can have an analog filter that outputs continuous principal component scores. And then we can look at principal component 1, which contains most of the power, and trigger the readout of all of the other principal components. And so in an event-triggered way, we can make a low-rank approximation of the incoming signal and look at a low-power representation of the spike waveform. And that reduces the total power in the system by a factor of about 60 relative to trying to transmit out and digitize the full data. It detects a spike, and then this is spike-based. Yeah. Field potentials are much lower data rate, so you can sample those without having to do fancy things. So as you can see from our recent interest in microECOG, We're interested in developing an array format that's a mix of penetrating and surface electrodes. Our first product will be essentially an intercalation of surface electrodes between penetrating probes that allows us to insert the device at reasonable density from a tissue standpoint and an insertion standpoint, while also getting out as much data as possible. And you could essentially, our device is a very pragmatic improvement upon the Utah array, which in our estimation was almost sufficient to become a medical device product, but lacked in probably 3 ways. One being that the information density is still a little bit low to deliver on applications. 2 being that it was never really packaged as a medical device. So if you see someone with a deep brain stimulator, you would notice that they have it until you look very closely. If you see someone with a Utah array, you would certainly conspicuously notice that they have it. And as a result of that portability, they're not used outside of the clinical trial setting. And then third, this aspect of materials. The Utah array coated in Parylene C, metallized with thin films to reduce the impedance. From a material standpoint, it's not really suitable as a class 3 device for medical implantation, which is why it has a 510(k) designation, is used successfully in clinical trials, but is probably unlikely to become, to receive pre-market approval as a medical device. But in some sense, all of those 3 things can be engineered around. The data rate can be increased, the form factor can be packaged like a DBS, and finally the materials, the reason why I talked a lot about microwires in the beginning is that microwires are robust. They're platinum iridium, they can be insulated with ceramics, they can be made of a material stack that can last 10+ years. And as long as they're small enough, and as long as they're inserted at the right density, they can last for a very long time and get signal for a very long time. So I'll just leave you with a little bit of a mock-up of the device that we're designing now. This device will be complete at the end of the year. We'll be doing some in vivo testing in sheep in early Q1 of next year. And I would anticipate that this system could be in an early feasibility study